Hello. Today we are going to talk about chapter 16, where we take everything from the nervous system that we've learned and we're going to put it all together into how everything functions as one piece. So, first thing we're going to do is talk about what sensation is. A sensation is that conscious or subconscious awareness. So, things that are happening outside of the body, that external environment, we're usually somewhat conscious to, but something that happens in the internal environment, most of the time we're not conscious to. So that's a subconscious awareness. Like we're not technically conscious to how fast something is moving through your digestive tract, how fast your heart is pumping. We're not technically, we're subconscious to that. So that type of sensation and the type of reaction is going to vary just depending on where these impulses are coming from. So that sensory information could be from reflexes, those spinal reflexes. Anything that reaches the lower brainstem is going to cause a more complex reflex. Changes in your heart rate, changes in your breathing rate, those are a more complex reflex. It's going to that lower brainstem. So if it's going into that lower brainstem, we're not aware of it. That's where that subconscious again comes into play. If something reaches the cerebral cortex, that is when we become consciously aware. So somebody touches you, you have pain somewhere in the body, you hear something, you taste something, you smell something. As long as it goes to the cerebral cortex, we are consciously aware that it is happening. If it does not reach that cerebral cortex, we are not consciously aware. That is where perception comes in. That perception is that interpretation of that sensation. Again, it has to come to the cerebral cortex for us to know that it happens. And then that interpretation is done within the cerebral cortex. If it does not reach that cerebral cortex, we are not aware of it. We have no perception that something is happening. Like the pressure of blood vessels, we are not aware of that. That goes to the medulla. So changes in blood pressure, we're not consciously aware that it is happening. So these different sensory modalities. We have a lot of different sensory modalities. Touch, pain, vision, hearing, these are different types of sensory modalities. And a sensory neuron can only carry information for one sensory modality. Test question. Sensory neurons that relay information for touch, they're not going to send information for pain. There's a different sensory receptor and neuron for pain than for touch. Just like there's a different sensory neuron for tickle versus hearing. They're different sensory neurons. So our general senses, those are those somatic and visceral senses. So those somatic, this is what is happening at the skin level. Touch, pressure, vibration, itch, and tickle. There's also thermal, so that hot or cold. There's pain and proprioceptive sensations. Proprioceptive sensations is where your joints are in space. So that position of your knees right now, the position of your ankles, the position of your hips, what position your head is in, these are all different proprioceptive sensations that allows your brain to know where your body is at. And then we have different visceral senses. These visceral senses are going to send information about to these internal organs. So pressure, stretch, chemicals, nausea, hunger, temperature. These are all visceral senses. And some of these things we have awareness of. Sometimes some pressure or stretches in the intestines can cause some cramping type of sensations. Nausea, hunger. We usually have conscious awareness of those. Then we have our special senses. Chapter 17. Smell, taste, vision, hearing, and equilibrium, which is the same thing as balance. So sensations, as we've mentioned, when I talked about reflexes, this is the same kind of concept. They're all going to start at sensory receptors. These sensory receptors can be their own specialized cell. They can be dendrites of a sensory neuron. These sensory receptors are going to respond to that one specific type of stimulus that they pick up. And that's where that selectivity is going to come into play. These sensory receptors, they can respond weakly or not at all to other stimuli. So they pick up their one main stimulus. They may pick up something else like pressure versus vibration, depending on how that vibration is on your skin. It can also technically pick up some of that pressure along with that vibration because that vibration could be pushed down into your skin. So it's also picking up the pressure of that. So when we get this sensory information, something needs to stimulate that sensory receptor. It has to happen in that receptive field, meaning it needs to be in an area where that sensory neuron that picks up that type of stimulus is located. If this stimulus is not in a receptive field in an area where that sensory neuron is located that picks up that specific sense, that information is not going to go to the brain. So we're going to have transduction of that stimulus. As in, it's going to convert the energy of that stimulus into a graded potential. That is known as transduction. From there, as long as we reach that threshold, those graded potentials reach that threshold, it can trigger one or more nerve impulses or action potentials. It's going to make its way into that spinal cord and up possibly up to the brain, depending on what it is. We need integration of that sensory input. We need to process that information. So when that information comes into that central nervous system, we need to process it. So conscious sensations or perceptions are done at the cerebral cortex. If it's not done at the cerebral cortex, we don't know it's happening. Just like you see with your eyes, you hear with your ears, you feel pain with that injured part of your body. So we're feeling this information because it's going up to that primary sensory cortex in the brain, that cerebral cortex. So we mentioned some of these different receptors. We have free nerve endings. These were mentioned also back in the integumentary system. So these are dendrites that don't have any kind of specialization to them. These are going to pick up that pain, temperature, tickle, itch, and some touch sensations. There's encapsulated nerve endings. So their dendrites have some sort of connective tissue capsule on them. They're going to pick up that pressure, vibration, some touch as well. There's different types of capsules. Some of them are going to enhance that sensitivity, or it's going to allow that nerve to better pick up that sensation because it's usually larger than that free nerve ending. So it gives a larger space or surface area to pick up that type of sensation. Sometimes we even have separate cells. These cells are going to come together with one of those first order neurons. An example, hair cells that we use for hearing and equilibrium in the ear. We have those gustatory receptors, which are taste buds, and photoreceptors in the eye. All of these are special receptors that attach to these sensory neurons to send this information into the brain. So these receptor potentials, they respond to a stimulus by generating graded potentials. So there's usually these free nerve endings or encapsulated nerve endings. So if that graded potential reaches that threshold, it can send those nerve impulses. Then those nerve impulses or action potentials will make their way up to the brain. If we get some graded potentials, but they're not large enough to generate that action potential, it's not going to make its way up to the brain. So here's a good example. We have a sensory receptor. We have something pushing down, putting pressure on that sensory receptor. This is going to take this information into the spinal cord, up to the brain, to that primary sensory cortex. That primary sensory cortex is going to let you know, okay, you feel that pressure and where you feel that pressure. So a first order neuron. First order neurons is the first neuron in a series. So here, this neuron is picking up cold. I go outside in the winter. It should be cold. Should be, at least in the Chicago area. That cold is going to create graded potentials. And when those graded potentials add up enough, they cause these nerve impulses to occur. And that information is going to make its way all the way into that central nervous system, to the brain. That's what lets you know it's cold outside. Vibration. Vibration, we'll play with tuning forks in lab so you can kind of feel what vibration feels like, but also running your fingers across a textured blanket, a textured sweater, carpet on the floor. Those are all create kind of like this vibratory type of stimulus. So this vibratory stimulus comes in. We have this encapsulated nerve ending here. So you see this enlarges that surface area a lot. So once we get a stimulus in, this stimulus is strong enough to reach threshold. This information gets sent off to that central nervous system to let you know you're feeling this vibratory sensation and where you're feeling it. Now let's look at taste receptors. We talked about special senses already. So this is a specific receptor that is used to pick up taste. So this one is picking up sugar, something sweet. So there's enough sweet to generate this nerve impulse. We're going to release neurotransmitter between this gustatory receptor and this neuron, this first order neuron that is going to take this information and send it into the brain. So they work just the same way, having these sensory receptors, but there's just this receptor that's going to attach to another neuron that's going to send this information off into the brain. So our receptors are kind of named based off of where they're located, what they do. So let's look at some of these. We have exteroceptors. Extero should tell you that it is at the out. It's picking up something from the outside of the body. So this is that external environment. So these exteroceptors are going to pick up hearing, vision, taste, touch, pressure, vibration, temperature, and pain, because this is all something that is at the external surface of the body. Interoceptors, also known as visceroceptors. So this is internal. So we're getting something from the inside of the body. So our blood vessels, we need to know the stretch put on our blood vessels, for blood pressure, those visceral organs, muscles, nervous system. So they're monitoring what is happening inside the body. They're not consciously perceived unless we feel that pain or pressure. These are the types of internal sensations that we usually feel. The next one, we have proprioceptors. I have mentioned these several times now. Proprioceptors are in muscles, joints, and tendons. So part of proprioceptors, besides knowing where your body is in space, they're actually going to help to give information on that equilibrium or balance. Body position, muscle length, and tension, and that position and movement of your joints. This is all coming from proprioceptors. This page goes through these kinds of receptors again based off of the type of stimulus. So mechanoreceptors. So deformation, stretch, or bending of cells. So these cells, these cells get stimulated, that stimulation changes the shape of these mechanoreceptors that causes these action potentials to be sent, such as touch, pressure, vibration, proprioception, hearing, and equilibrium. They also monitor the stretch of blood vessels and internal organs. Thermoreceptors. Thermometer. Temperature. Nociceptors. These are specific to pain. It could be physical damage. It could be chemical damage that triggers these nociceptors, sorry. Photoreceptors. They're going to detect the light that hits the retina, that back of the eye. Chemoreceptors. Chemochemicals. So, taste, smell, and bodily fluids. We need to know how much oxygen and carbon dioxide is in our system, how much calcium is in our blood, how much of all these different ions to help try to maintain and regulate all of this. Then we have osmoreceptors. Osmotic pressure. So that movement of fluids along with the different proteins in the blood and the pressures that are being put onto the blood vessels versus that pressure of fluids leaving the blood. You'll go a lot more into this osmotic pressure and anatomy too. There are test questions between both of these slides, all the different types of receptors. So our receptors go through adaptation. So when you're sitting next to somebody who is wearing very strong perfume or cologne, you smell that perfume or cologne for a while, and then eventually you don't smell it anymore. That is adaptation. So if we have a constant stimulus, those receptor potentials decreases the amplitude. So it filters things out so you can focus on something else. So there's that frequency of nerve impulses decreases. So the perception of that sensation can fade. It can even disappear. Like that cologne. Another example, this is the example from your textbook, is when you step into a hot shower. The water can feel really hot at first, but then your body adjusts to it. Even though the temperature of the water does not change, you kind of adapt to that hot temperature to take a hot shower. We have different types of receptors that adapt. We have rapidly adapting. They do just that. They adapt very rapidly. So these are for signal changes in the stimulus, like vibration, touch, and smell. This is why if something were to stay on your somewhere vibrating, eventually your body would kind of filter that out. Touch. Somebody was touching you, putting pressure on you, eventually that would get filtered out. Same with that smell that I mentioned. And the difference is we have slowly adapting receptors. These either adapt slowly, they will continue to send these nerve impulses as long as that stimulus persists, though. Pain. body position, chemical composition of the blood. Some of this is information that we always need to know. Pain, I mentioned highlighted pain very large because I mentioned touch before. If somebody was touching you, but it was painful, that stimulus isn't going to get filtered out. Like if you go get a massage and somebody's pushing down on a trigger point or a knot in your shoulder, your back that really hurts, that pain's not going to go away until that knot goes away. So even though touch can be rapidly adapted to, that pain that could be created from that touch will not. So this is a nice chart from your textbook that goes through the different types of receptors, the classification based off of location, and that classification based off of what type of stimulus. These are nice little review charts of all of this information. So, somatic sensations. These come from stimulation of that sensory receptor in the skin or subcutaneous tissue. It could be in the mucous membranes of the mouth, vagina, anus, skeletal muscles, tendons, joints. These somatic sensations, these receptors are distributed pretty unevenly. Like in your hands, receptors are very close together, but on your back, they're a lot further apart. There's this thing called 2-point touch where I have some cards, I'll try to remember to bring them in for lab, that have 2 points on them, and you can kind of figure out how far sensory receptors are. But like you take something and you touch somebody in 2 different parts at the same time. You ask them how many they feel. You move them closer and closer until they only feel 1. In your back, it's going to be a lot further apart. On your hand or your fingers, it's going to be a lot closer together before you only feel 1, even though there are 2 things touching you. So it's kind of fun to play with, to kind of see how far apart sensory receptors are in different areas of the body. So, as I mentioned the hands, your tongue is also a place where there is a lot of sensory receptors close together. Same with your lips. So, cutaneous sensations, cutaneous because our skin is a cutaneous membrane, if you remember back to the beginning of the semester. A cutaneous sensation comes from that skin surface specifically. Those are part of these somatic sensations. Tactile sensations. Tactile is touch, pressure, vibration, itch, tickle. These are different types of sensations that tactile types of receptors are going to pick up. These differences in these types of sensations, they do come from the same type of receptors. Most of them are encapsulated mechanoreceptors. They can be large diameter, medium. They're going to pick up that touch, pressure, vibration. Sensations like itch and tickle. Those are free nerve endings. We've got tactile receptors in the skin. We have tactile corpuscles, hair root plexuses, non-encapsulated sensory corpuscles, lamellar corpuscles, bulbous corpuscles, and free nerve endings. These are all going to pick up these different types of tactile sensations. So there's lots of different types of nerves and nerve endings that are going to pick up these different types of sensations. So touch. This results from the stimulation of tactile receptors in the skin or subcutaneous membrane. Some of these tactile corpuscles are known as Meissner's corpuscles, is the other name. That's the name I learned them as. They are in the dermis of hairless skin, palms of your hands, the bottoms of your feet. These are egg-shaped masses of modified Schwann cells. They're rapidly adapting. So you're going to feel this sensation, but if this sensation continues, it's eventually going to kind of fade off. We've got them in our fingertips, hands, eyelids, tip of the tongue, lips, nipples, soles of your feet, clitoris, and tips of the penis. These are all technically hairless areas. These hair root plexuses, they're also these rapid adapting that are found on hairy skin, because hairy skin, hair, all hair has these nerve plexuses that wrap around them. They are free nerve endings. They're going to detect movement that causes the hairs to move specifically. So we looked at this back in chapter 5 at the integumentary system. We're back because we're talking about the nerves specifically. These wrap around this hair follicle. Here you can see a tactile corpuscle. You can see a bulbous corpuscle, which we're going to mention. These non-encapsulated sensory corpuscles, we've got free nerve endings over here. And then we have lamellar corpuscles. They all pick up something different, like these free nerve endings have this pain, itch, tickle, cold, warmth. These non-encapsulated does continuous touch and pressure. These tactile are going to do the onset of touch and low frequency vibrations. These bulbous are going to stretching and pressure. These lamellar do high frequency vibrations. So there's a lot of different things that these are going to pick up. So here's more. We mentioned them on the slide before. So these non-encapsulated sensory corpuscles or Merkel's discs. They are kind of this saucer-shaped, flattened free nerve ending, and they're going to come in contact with those tactile epithelial cells. So they're in that stratum basal specifically. So fingertips, hands, lips, external genitalia, touch. If you're holding something for an extended period of time, they're going to respond to that, so you know. Then we have those bulbous corpuscles or Ruffini corpuscles, same thing. These are in the dermis, subcutaneous tissue. They are branched nerve endings and Schwann cells. They're very sensitive to stretching. So if a massage therapist stretches your skin, these bulbous or Ruffini corpuscles are going to pick up that stretching. Pressure is a sustained sensation that is felt over a larger area because you're pushing down harder. For pressure, you push down harder, so it tends to be felt over a larger area just because of that deeper deformation of the skin. So lamellar and bulbous corpuscles are going to respond to this pressure. They respond to that steady pressure. They're slow adapting, so they're going to continue to let you know that something is touching you. That vibration. Rapidly repetitive sensory signals. So these lamellar corpuscles or Pacinian corpuscles, same thing. They have this multi-layered connective tissue capsule that somebody decided looked like a sliced onion. They adapt very rapidly. Dermis, subcutaneous tissue. These lamellar specifically do high frequency power drill, electric tools, if you've ever used any of those, tactile corpuscles. They also detect vibrations, but they do low frequency. So as I mentioned, moving your hands across a textured object like a blanket or even a paneled type of door, every that that sensation as you move your hand over that paneled kind of wall or door, that's technically a type of vibration. Itch. Itch is a stimulation of free nerve endings by chemicals. Bradykinin, histamine, or even the antigens from mosquito saliva when you get a mosquito bite. Scratching can usually alleviate the itching because what scratching does is it causes the pathway that usually sends this itch signal into the spinal cord, into that central nervous system, to be blocked. That's why when you scratch an itch, it stops, usually. That mosquito saliva, depending on how you react to mosquito bites, it may not alleviate that itch, or it'll temporarily alleviate that itch for a short period of time. But scratching that itch does help for short periods of time. Tickle. Tickle is an interesting one. We think that free nerve endings. So, tickle can only arise when someone else touches you. Why? Because your brain always knows where your body is in space. Where your hands are at all times, your body knows. So, knowing where your body is in space, you're not able to tickle yourself. Only when somebody else touches you unaware will it stimulate these nerves that send this type of tickle sensation. Here's an interesting topic. There's some really good books out there on this and different ways to work with people who have this phantom limb sensation. So somebody who's lost an arm or a leg, they can still experience these different sensations in the limb that's not really there. So they may feel this itching sensation in their arm that was amputated, pressure, tickling, or tingling, pain, because when you lose that limb, those nerves that went to that limb are severed. Those severed nerve endings can get activated. And then your brain kind of takes that information and sends out that there's this itching on an arm that's not there. There's lots of explanations for this. There's lots of really cool research out on this. There's another thought that cerebral cortex has undergone some functional reorganization after that limb has been removed. and it allows it to respond to stimuli from another body part. So this remodeling could give that idea of false sensory perceptions from that missing limb. And this phantom limb pain, people report some pretty severe, intense pain. And pain medications don't work because that limb where they're feeling pain really isn't there. So there's some different electrical nerve stimulation techniques that they can use. Acupuncture. I've read some acupuncture on this. It's kind of cool. Biofeedback. Some of these biofeedbacks, there's an entire book on using like a mirror box, like a box with a mirror in it, and you put the limb that you do have in that box and use that mirror to kind of trick your brain into thinking that what it sees in that mirror is the limb that was amputated. That's no longer there, but your brain sees that it's there through this mirror, in this mirror box. And then there's a lot of different kind of treatment techniques that they can use within that to help the brain retrain that brain that limb really isn't there, going through this whole process. It's really cool. So let's go back to different types of receptors and sensations. Thermo, temperature. Thermoreceptors have a 1 millimeter diameter, receptive field all over the skin. It's kind of cool. So cold receptors are found in that stratum basale, that bottom layer of that epidermis, and they actually pick up temperatures between 10 to 35 degrees Celsius or 50 to 95 degrees Celsius. That is what cold receptors can pick up. Warm receptors, we don't have as many of those. Again, they're in the dermis, and they pick up anywhere from 30 to 45 degrees Celsius, 86 to 113 degrees Fahrenheit. They are rapidly adapting. You go outside when it's really hot. After a while, you don't necessarily feel as hot as it was when you first took that step outside. But they're still going to continuously send this information. So anything below, this is an interesting one, below 50 degrees Fahrenheit and above that 113 degrees Fahrenheit actually stimulate pain receptors. So below 50 degrees outside can actually feel more like pain. Above 113 degrees can feel like pain instead of hot or cold. Something kind of interesting. So pain. Pain is thought, it is a protective type of function. And there's pain signals get sent on these nociceptors by tissue damage or tissue conditions. So these nociceptors that pick up this pain, they're those free nerve endings. They're found in every tissue of the body except the brain. This is why when they do surgery on the brain, people are usually awake for it and they don't feel it. Once they get past the skin and the bone to get into the brain, you don't feel what's happening in there because there are no Nociceptors, there's no pain receptors in there. So that intense thermal, that hot or extreme hot or cold, mechanical or chemical stimuli will activate these, and prostaglandins, kinins, and potassium can stimulate these nociceptors, and these can get released when tissue is damaged or there's some sort of injury in the area. Pain, even if the stimulus that caused that pain was removed, those chemicals tend to linger in that area, which causes pain, that sensation of pain, to be felt longer than the actual stimulus that is causing the pain, the initial pain, to appear. So these nociceptors really do not adapt. That's why when you're in pain, you always know you're in pain. So excessive stretching can cause pain, prolonged muscle contractions, muscle spasms, or not enough blood flow to certain area and organ. These can all stimulate pain. So we have different types of pain. Fast pain. It happens very fast. Within 1/10 of a second, we feel that pain. This is acute, sharp, or prickling or pricking type of pain, like a needle. We don't feel it in deeper tissues of the body. It's usually more superficial. And then we have slow pain. This takes a couple seconds to feel after the stimulus to actually feel it. It's going to continue to increase in intensity. Chronic, burning, aching, or throbbing pain is carried on these types of nociceptors. It can be in the skin, or it can be deeper tissues or even internal organs. So superficial pain. Those of you going into the medical field are going to deal with pain a lot. That's why this chapter talks about it. Is stimulation of those receptors in the skin. Deep somatic pain. Somatic tells me it's going to be skeletal muscles. So it's receptors in those muscles, could be those joints, tendons, or fascia. Visceral pain. Visceral pain again tells me right where it's coming from. Those visceral organs. Fast pain is usually very local. They can pinpoint exactly where that pain is. Slow pain, it can be very localized, but it's more diffuse. So it involves a larger area. They kind of draw a circle or kind of say this area is where they feel the pain instead of fast pain. They're like right here. They're able to point to something and say this hurts and point to exactly where it is. Versus that somatic pain, they kind of give an area. Visceral pain, usually in or deep to the skin, and they actually feel it over that organ, or there's what we have as referred pain. Referred pain is somewhere far from that stimulated organ. So here's this nice picture that shows us where certain types of pain are felt. Most people know about the heart being in that left chest, down that left arm. Here is how this referred pain works. So, when an organ that has pain is sending this information, it's going into the spinal cord. This carried on the same nerves that are going into the spinal cord where that organ is sensory information that goes to the skin, to that same level or same area. So here it says the sensory fibers from the heart, the skin superficial to the heart, and the skin along the medial aspect of the left arm, so that T1 to T5, they all go into the spinal cord at the same segment. So as this information goes up to the brain sometimes can get a little confused on, was this sensation coming from the arm? Was it coming from that chest area? Was it coming from the heart? Because these are all information coming in at the same place. So people with kidney pain can experience low back pain. Someone with liver and gallbladder pain can have right shoulder pain. Feeling pain in the jaw for the heart, that is more of a sensation that women feel. Men usually feel down that left arm. Women usually feel it going up into the face. It's interesting. Men and women experience a heart attack very differently. The small intestine that's listed here, this is more the appendix. Someone who has appendix problems, that usually starts in like a ring around that umbilicus, and then it'll travel down to that right lower quadrant where that appendix sits. So proprioceptive sensations. It allows us to know your arms are yours, your legs are yours. Let us know where our head and limbs are. Hopefully your head's attached to your neck. Your arms are attached to your body. You're able to know this even if we're not looking at them. Kinesthesia. Perception of body movement. We're able to know where our body is moving without looking at it. Proprioceptors. That proprioceptive sensation that's in our muscles and our tendons and lets us know how far a muscle is contracted, allow us to know how far your knee is bent, how far your ankles are bent, how far your hips are bent, to allow you to put together a movement plan for when you want to get up and walk out. Go from sitting to standing without falling over. These proprioceptors allow us to know this information. Weight discrimination. Allow us to pick up something and kind of assess the weight of it, kind of have an idea of how much something weighs. It allows you to even look at something and determine how much muscle strength it's going to take to pick up your computer versus picking up your pencil. We need more muscle to pick up a computer than we do to pick up a pencil. We need more muscle to pick up a box of books than you would to pick up your pen. So just looking at it lets you know how much it's going to take to pick something up. We mentioned muscle spindles before. So these muscle spindles are part of those proprioceptors because they're detecting that specific length of your skeletal muscles and how much of they help with those stretch reflexes. So that patellar reflex that everybody should know. We're able to maintain and know just how much these muscles are contracted, how much our muscles are stretched. The brain does set this muscle tone, which is this partial contraction that is present when our muscles are at rest. So, right now, your muscles have somewhat of a contraction in them while you are just sitting wherever you might be sitting or laying down. Just holding your head up, you need some sort of muscle tone there to help pick and hold your head up. These intrafusal fibers, we have several slowly adapting sensory nerve endings that are going around these muscle fibers. We looked at them when we looked at reflexes. So these intrafusal fibers are in here. We have these intrafusal, we have extrafusal muscle fibers, we have these sensory nerve endings. These are all working to send information into the brain on how far these muscles are contracted or how far these muscles are stretched. So we have them in most of our muscles in our body. We have more in muscles that allow us to do fine, controlled movements, like your fingers, your eyes. And if anybody's ever played an instrument, those muscles that are being used to play that instrument have a lot more muscle spindles. So those muscle spindles are telling us that specific length of that muscle, whether it's stretched, whether they are contracted. This information is being passed along on those muscle spindles. This information is constantly being sent into our brain. This information, it goes to that primary somatosensory cortex. So we have this conscious awareness and control over where our limbs are at and how they're moving. But we're also sending this information into that cerebellum. This is what helps us to coordinate our muscle contractions. So when you go to pick something up, not only are we making sure that your biceps contract, your triceps relax, but we have enough of those bicep fibers, maybe that brachialis underneath that biceps brachii needs to contract. Maybe that brachioradialis needs to help. Those are all forearm flexors. All of these different muscles are working together to allow you to move. Your muscles all contract at this right time, at the same time, to allow you to pick something up. Gamma motor neurons, they're going to terminate by these intrafusal fibers. They work to adjust the tension on those muscle spindles. So I've given this example. When your biceps muscle shortens, when you lift something up, this gamma motor neuron stimulates these fibers to contract slightly to keep those intrafusal fibers kind of taut and the contractile muscles surrounding that spindle They're working to reduce that tension. So those frequency of impulses is going to tell us or tell that muscle how much that is stretching, how much this muscle is contracting. They are a lot more sensitive to stretch these gamma motor neurons. Then those extra fusel muscle fibers, these are alpha motor neurons. So these gamma and alpha neurons come from the anterior gray horn of the spinal cord. So during a stretch reflex, that muscle spindle, those sensory neurons, are sending this information from that stretch. Hitting that patellar tendon causes a slight stretch of that muscle. It sends that information into the spinal cord. It goes into the brain, so you know what is happening. It sends that information out on that alpha motor neuron. It's going to go to that extrafusal muscle fibers of that same fiber, causing it to contract. And that relieves that stretching that happened when that reflex hammer hit that patellar tendon. So I showed all of that. Tendon organs. We kind of mentioned those are in the with the reflexes too. So these tendon organs are slow adapting receptors. They are found at that junction of that tendon and that muscle. So if we do these tendon reflexes, those tendons want to protect, those tendon organs specifically want to protect those tendons and the muscles they are attached to. They're trying to prevent damage from excessive tension. Each of these tendon organs has a thin capsule of connective tissue within these tendon fascicles. So here's this tendon organ up here. So all of these little sensory fibers here are sending this information into the brain on what is happening within this tendon organ. So when tension is applied to a muscle, that tendon organ is going to send information to the brain to let that brain know there's changes in the muscle tension that that tendon organ is attached to. So that tendon reflex can help to decrease muscle tension by causing that muscle to relax again. So that's why that patellar reflex, that quadricep contracts, your leg kicks out, but then it relaxes back down. We have joint kinesthetic receptors. These are at articular capsules of synovial joints specifically. So all those freely movable joints that have that synovial capsule on it, we have free nerve endings and bulbous corpuscles found there. They respond to pressure at those joints. So they're going to respond. We have small lamellar corpuscles that respond to acceleration and deceleration of that joint during movement. So running, you speed up versus slowing down. That small lamellar corpuscle is responding to that. Those ligaments at that joint contain, similar to a tendon organ, they're going to help to readjust this reflex to help prevent damage to those tendons, to those ligaments, to the muscles in the area. So they're also playing into these different reflexes. When different strain is put on different joints, this information is going into that brain. So here's some nice charts that go through the different receptors, where they're located, how they adapt to sensations, and where they're found. Located, found, same thing. So let's look at these different pathways. We mentioned first order neurons. Here's where we're going to go into more detail. So the somatic sensory pathway, we've kind of talked about this already, but this is taking that information from different sensory receptors around the body to that primary somatosensory cortex. This is in that parietal lobe of the cerebrum, but then it also sends this information into the cerebellum. So we have first order neurons. This is the first neuron in a series of neurons. This is the neuron that's going to take these nerve, they're going to conduct those nerve impulses specifically from those somatic receptors. So these first order neurons are located outside the central nervous system. First order neurons are found outside the sensory nervous system. Second order neuron. This is going to take information, going to take it from this brainstem, can take it from the spinal cord. It's going to conduct this information into the thalamus. The thalamus is kind of that main relay station. So it's going to connect to a third order neuron. But here's some interesting thing. Within the second order, axons of the second order neuron are going to desiccate or cross over. As I had mentioned before, the right side of the body is controlled by the left side of the brain. The left side of the body is controlled by the right side of the brain. So this information needs to cross over in the spinal cord or the brainstem somewhere. It's usually at the brainstem, but some of it does cross over at the spinal cord before going to the thalamus, where we get our third order neurons. They're going to take information from the thalamus to the primary somatosensory cortex on the same side at this point from where it came into the thalamus. So if it hits that somatosensory cortex, that is when we're aware of whatever type of sensation we could be feeling. Information is always perceived by the opposite somatosensory cortex from where we feel it, where that initial sensation took place. So somebody touches your right shoulder, the left side of your brain is going to process that. If somebody touches your left shoulder, the right side of the brain is going to process that. So different relay stations. I kind of mentioned that thalamus as being kind of a relay station, but this is an area in the central nervous system where neurons are going to connect to other neurons of sensory or motor, and they're being relayed from one area of one region to another in that central nervous system. So the thalamus is the main relay station of the brain. Test question. Even though the spinal cord and the brainstem can still can relay information, that thalamus is the main relay station. So sensory information, we kind of mentioned some of these pathways, but their information is going to go into the thalamus from the posterior column medial lemniscus pathway, the anterolateral pathway or spinal thalamic pathway, and the trigeminal thalamic pathway. So these are different ways that this information is going to come into that thalamus. And again, it usually tells you two of the three names at least tell you where they're located. So this posterior column, medial lemniscus, anterolateral and trigeminal thalamic pathway, it is taking information into that primary somatosensory cortex in that parietal lobe of the brain. These pathways cross over in the brainstem or the spinal cord. So information brought in by these three pathways are perceived on the opposite side of the brain from where it happens. The posterior column medial lemniscus pathway. This is bringing touch, vibration, pressure, proprioception into that cerebral cortex. Our first order neurons are found in that periphery. It's going to take this information into the spinal cord. It's going to take this information all the way up to the medulla. In the medulla, they're going to synapse with second order neurons, where they're going to cross to the opposite side and enter the medial lemniscus. At the thalamus, they're going to synapse with third order neurons that take them to the primary somatosensory cortex of the cerebellum. So let's look at some of these. Here's that posterior column medial lemniscus pathway. So this sensory information is coming in, goes in through the posterior roots, comes in, and then it's going to jump to those posterior columns that are labeled right here, that are shown in blue, before it travels up to the medulla. This is still the same neuron that was out in the periphery that felt that initial sensation. Now we've made it to the medulla. From here, we're going to switch sides, cross over to the other side of the spinal cord or that brainstem, make our way up to the thalamus, where this information is going to go off to that primary somatosensory cortex to allow us to feel whatever type of sensation was felt there. We also have the anterolateral or spinal thalamic pathway. Oh, I'm just going to go straight to pictures. So this sensory information that comes in on this anterolateral pathway or spinal thalamic comes in through the spinal cord. It's going to synapse right away with a second order neuron that's going to cross the spinal cord over and travel all the way up to the thalamus. Now we've got our third order neuron taking us into that primary somatosensory cortex. So, in other words, some of this information crosses to the opposite side in the spinal cord. Some of it crosses over in the brainstem. Either way, it's still going to be processed by the opposite side of the body. The last one is the trigeminal thalamic pathway to the cerebral cortex. So pain, temperature, touch, and proprioception from the face, mouth, and teeth. So this information is coming in, actually, into the pons. It can either travel down to the medulla, where it crosses over and travels up to that thalamus, or as it comes into the pons, it can cross over and travel up, where they're going to go to that primary somatosensory cortex, so we know where something touched us, touched our face. So that primary somatosensory cortex, we've talked about it since chapter 14. This is the post-central gyri of the parietal lobes of that cerebral cortex. So how do we know where, or how does our brain know where we felt something? We have what is known as a sensory homunculus. On this sensory homunculus, we have a map of the different body regions. So, the size of that cortical region that's going to represent a specific body part, it can expand and take up a larger area. It can also shrink depending on how many sensory impulses are coming in from that specific body part. So let's look at this. This is the primary somatosensory cortex on that postcentral gyrus of the parietal lobe. You can see here, it kind of lists off all of the different areas within this sensory cortex and then where we feel it in the body. So, something touches your hand, it is going to come up and it's going to go to wherever you felt that sensation on your hand. Something touched your forearm, going to come up to that specific area in that sensory cortex so you know something touched your hand. Something touched or you feel pain in your teeth. Well, that's going to come into this general area. So these different areas or cover that specific area in the body. So when something touches you at that specific area, it's going to go up to this sensory homunculus, to this specific space, and go right into where this specific area is in the body. So you know. So let's look at, we have somatic sensory pathways that go specifically to the cerebellum. So we have anterior spinocerebellar tract, the posterior spinocerebellar tract. So things that need to go into the cerebellum is information for posture, balance, and coordination of different skilled movements. So these are nice charts from the textbook that go through these different pathways and how this information, where this information is going into the spinal cord and where it is traveling up to the brain. So now let's look at the motor side. The motor side helps to control voluntary movement. It's either excitatory or inhibitory types of signals that is going to control that movement. So we have lower motor neurons. All a lower motor neuron means is their cell bodies are in the lower part of the central nervous system, brainstem, spinal cord. So from that brainstem, lower motor neurons can go through those cranial nerves to go to those specific skeletal muscles for the face and the head and the shoulders. From the spinal cord, those lower motor neurons are going to go through spinal nerves that go to skeletal muscles. Lower motor neurons are going to provide that output from the central nervous system. So they're that final pathway to that specific movement. Upper motor neurons. Upper motor neurons are found in that upper part of that central nervous system, and they help to regulate the activity of those lower motor neurons. They're actually interneurons. Interneurons are those in between neurons, but they're still, since they're relaying motor information, they're kind of called those upper motor neurons. So we have 4 different types of neural circuits that are going to help control your movement. Local circuit neurons. So this information goes into those lower motor neurons that are by interneurons that are located close to those lower motor neurons. So we're going to receive that sensory input to help give us some sort of controlled movement, flexion, extension, depending on what type of sensation is felt. Our upper motor neurons. They have a local circuit and a lower motor neuron. They receive information from those upper motor neurons. So those cell bodies work as processing centers. So most upper motor neurons synapse with local neurons, which synapse with lower motor neurons. So some of these upper motor neurons can synapse directly with lower motor neurons. These upper motor neurons come from mostly that primary motor cortex, and they're helping to plan and execute movement. Some of them can originate from the brainstem. Those regulate your posture, your balance, your muscle tone, reflexes of your head and your trunk. Like you hear a noise over your shoulder, you turn your head. That's more of a reflexive type of movement of the head. Corpus striatum neurons. These assist in movement. So these neurons connect through the corpus striatum. They're going to connect with motor nuclei in that thalamus and that brainstem. These help to initiate and terminate movement. They suppress unwanted movement. They establish a normal level of muscle tone. So if they initiate a movement, you're sitting down, you want to stand up, that first part of the movement to stand up, that's initiating that movement. You want to take a step to walk, start walking. How you pick your foot up to move, that whole plan of how to do it is sent out through these corpus striatum neurons to initiate that movement, to initiate that walking. Then when you want to stop, they're the neurons that come in and stop that movement, suppress unwanted movements, your legs moving to the side, your hands kind of tremor, shaking as you move. These corpus striatum neurons are working to help create smooth, accurate movement. And then we have cerebellar neurons. They help aid your movement by controlling those upper motor neurons. So we've got neuronal circuits that are going to connect through that cerebellum because they're helping to kind of fine tune your movement. So that cerebellum is going to monitor the difference between intended movements and what movements actually performed. They're going to help to try to reduce errors in movement. They make sure your movements are coordinated. So you're stepping forward with your left foot, your right arm usually swings forward. Stepping forward with your right foot, your left arm usually swings forward. But they also help to maintain posture and balance. Paralysis. There's two different types of paralysis. Flaccid paralysis. This is where there's some sort of damage to those lower motor neurons. And since they're lower motor, they're on the same side of the body because they're the ones going out to those skeletal muscles specifically. So we don't have voluntary movement. There's no reflex action to those muscles that nerve goes to. Since that nerve is damaged or doesn't work, muscle tone is lost. So that muscle is limp. That limpness is known as flaccid. This is what happens when the nerve that goes directly to a muscle doesn't work. We also have spastic paralysis. This is where that upper motor neuron is involved. So that's supposed to help remove inhibitory influences. Does not happen, and this is on the opposite side of the body. Muscle tone is increased. Reflexes are exaggerated. You may even see some of those pathological reflexes, like that Babinski sign that we've talked about previously. So our premotor cortex. The premotor cortex takes the idea or desire to move your specific body part. They kind of generate this idea from one or more of these cortical areas. So they're taking information from the prefrontal cortex, the somatosensory area, the auditory, the visual area. All of this information is coming in to help plan out a movement. This information gets sent to the corpus striatum, where it's going to process this information, send it to the thalamus, then to that premotor cortex, where we develop a plan of how to do a movement. So we're figuring out which muscles should contract, how much they need to contract, and in what order they need to contract in. So you want to pick up a pencil, you want to pick up your phone. We need to create a muscle plan so you can reach for your phone, what muscles are contracting so you can reach, which muscles now, once you allow you to pick up or grab your phone, which muscles are contracting to allow you to pick it up towards your face so you can look at it. This is an entire plan that gets created. So this premotor cortex goes to the primary motor cortex. That's what allows us to execute this plan. Once it creates a plan, it also stores the information about this plan for next time. So when we activate the appropriate neurons, it causes those specific muscles to contract in a specific sequence to perform a specific activity. So that primary motor cortex, it allows you to execute voluntary movement. So electrical stimulation of any point of that primary motor cortex causes contraction of specific muscles on the opposite side of the body. Because again, right side of the brain controls the left side of the body. The left side of the brain controls the right side of the body. We are working to control these muscles by forming pathways through the spinal cord, through the brainstem, the spinal cord. But within this primary motor cortex, just like on the somatosensory side, we have a sensory homunculus. We also have a motor homunculus. So we have areas in this entire cortex that allows us to move each individual part of our body one way or another here. So this motor homunculus is where this information goes into that allows us to contract those specific muscles in a specific order to allow a specific movement. So we have different pathways of neurons that send out this information. Just like we have that sensory pathways for information to come in, we have motor pathways for information to go out. So in the direct pathway, it takes this information directly from that primary sensory motor cortex to those lower motor neurons to send out that information. Indirect. It does provide input to those lower motor neurons from those motor centers in the brainstem. Both of these pathways are going to send information into those lower motor neurons to allow those muscles specifically to contract. So voluntary movements are going to come from that primary motor cortex through direct motor pathways or pyramidal pathways. Those pyramidal cells are those upper motor neurons. We mentioned in chapter 12, they have that pyramid-shaped cell body. So the corticospinal pathway, going from the cortex down the spine, tells us where it goes. This is what allows us to move our limbs and our trunk. So those upper motor neurons in that primary motor cortex form this corticospinal tract that sends this information out. They're going to go through the internal capsule and the cerebral peduncle. At the medulla, they're going to go through the pyramids within that medulla. Here, they're going to cross. They're going to desiccate to the opposite side. And then they're going to go down. They're going to synapse with those lower motor neurons that are going to go out to those skeletal muscles. Only 10% of them desiccate at that specific spinal level where they go out. So most of them desiccate again in that brainstem. So we have a lateral corticospinal tract and an anterior corticospinal tract. The lateral corticospinal tract cross over in the medulla. Then they form that lateral white funniculus. They are going to synapse with those lower motor neurons in the anterior gray horn of the spinal cord. They're going to exit through those anterior roots into those spinal nerves that go to those specific skeletal muscles to produce that movement. The anterior corticospinal tract. These do not cross over in the medulla. So at each spinal cord level, these axons are going to cross over. They're going to cross through that anterior white commissure where they're going to synapse with their lower motor neuron. So here's this information in a picture. So this motor pattern is coming down through the spinal cord. They make it to the medulla before they cross over, travel through that lateral corticospinal tract where they're going to jump back into that gray horn to send out that information on that lower motor neuron. Versus the anterior corticospinal tract is the one that travels all the way down that spinal cord to its designated level, crosses over, jumps into that gray horn before it leaves on that spinal nerve, that lower motor neuron out to that specific skeletal muscle. The cortical bulbar tract or pathway. So this is what sends information for control of the head. So it's going to descend on that cortical spinal tract through the internal capsule. Some are going to desiccate, some are not. So these axons terminate at motor nuclei of one of the cranial nerves, the 9 cranial nerves that allow for motor movement, where they're going to synapse with those lower motor neurons or those cranial nerves. They're going to allow those specific movement. So those cranial nerves that allow movement, we have oculomotor, trochlear, trigeminal, abducens, facial, glossopharyngeal, vagus, accessory, and hypoglossal. Those are the ones that allow for specific movement. So they've got motor fibers, at least within that specific cranial nerve. So this information that's coming out, it's coming from that cerebral cortex going to whatever specific level and going out to wherever its destination is in the head or the neck. So our brainstem is going to do some control of movement as well. So let's go through some of these. We have indirect motor pathways, as we had mentioned before, those extrapyramidal pathways. This is for all somatic motor tracts outside of that cortical spinal and the corticobulbar. So we have rubrospinal, tectospinal, vestibulospinal, lateral reticulospinal, and medial reticulospinal. These cause involuntary movement for posture, balance, muscle tone, and reflexive head and trunk movement. So these different pathways are going to travel through, they're coming from the brainstem, they're going to cross over, and they're going to leave at their specific level to go to for their specific purpose. So this vestibular nuclei, it will help with postural reflexes. So it keeps your body upright and balanced. So your eyes, that vestibular apparatus in your ears, and your proprioceptors from your muscles and joints are all going to provide information to help with these postural reflexes. So your eyes are going to send in visual information about the position of the body in space. So what your eyes are seeing is going into that area. This vestibular apparatus is going to provide information about the position of your head, while those proprioceptors are going to send that information about the position of your limbs. Where is your arms and your legs in space? So in response to this information, we've got those upper motor neurons. They're going to activate those lower motor neurons, which are going to work with those postural muscles to keep your body upright. The vestibulospinal tract, this is information that's going down to skeletal muscles to help maintain that posture in response to different equilibrium changes. So when we talked about special senses, we kind of went into some of this different information about your balance, your equilibrium, your ears feeling what is happening outside, and how sometimes your skeletal muscles need to respond to that. So that's going to go down that vestibulospinal tract. The reticular formation is also going to help with posture. It can alter muscle tone. So it's going to receive information from your eyes, your ears, your cerebellum, that corpus striatum. it's going to take this information along either the medial reticulospinal tract or the lateral reticulospinal tract, and it's going to send information to skeletal muscles of the trunk and proximal limbs. So that medial reticulospinal tract does the trunk and extensor muscles, the lateral inhibits those trunk and extensor muscles. So one is going to excite it, the other one's going to inhibit. So contract muscles, prevent contraction. They work together because we need to maintain our posture and regulate that specific muscle tone during movement. That's superior colliculus. That is giving us visual input and auditory input. We have the superior and the inferior colliculus. They work together. So when input occurs in an unexpected manner, that superior colliculus is going to send information on that tectospinal tract. And it's going to activate muscles of the head and trunk to allow you to turn and look at something. So, as I said, turn in that direction of whatever stimulus was just induced. There's also that area for saccades, which is it's movement for when your eyes are reading, but it's also movement when a person looks at a different point in the visual field. So again, this information going into these different gaze centers in that reticular formation, and it's allowing the eyes for horizontal and vertical saccades. So as you're reading from left to right, your eyes are moving from left to right. There is a way to bring out saccades in somebody. You put them in one of those chairs that spin and spin them really fast, and then you stop, make sure their eyes are open, and you look at their eyes, and their eyes kind of have this tick going to them to the side. That's inducing like these saccades in somebody. People who get vertigo will have these saccades. If you move their head in a certain position, their eyes will start kind of ticking in this rapid sensation motion, and those are known as saccades. The red nucleus. The red nucleus will send information on the rubrospinal tract, rubro for red, and it's going to activate skeletal muscles for fine, precise, voluntary movements to the distal parts of your upper limbs, aka your hands. So that corpus striatum works to initiate muscle movement, suppress unwanted movement, regulates your muscle tone, and regulation of non-motor processes. So it's going to take in information. I mentioned all of the different parts to initiating a movement, but it's going to take in that sensory information around it. It's going to associate it, go to those motor areas, go to that corpus striatum, the thalamus, the premotor cortex, the upper motor neurons, and that primary motor cortex, the corticospinal and the cortical bulbar to promote that movement. It'll inhibit neurons of the thalamus that affect activity of that upper motor neuron. Going to go through that reticular information and reduce muscle tone. And it's also taking information for sensory, limbic, cognitive, linguistic types of functions. So there's different disorders of the corpus striatum. And these are going to affect body movement, cognition, and behavior. One such disorder is Huntington's. This is an inherited disorder. It affects that caudate nucleus and the putamen that degenerate, and it has to do with GABA or acetylcholine. These people have chorea, which is rapid, jerky movements that are involuntary and there's no purpose to them. They also they get mental deterioration. So they're losing their memories. This usually doesn't start until 30 to 40 years old, and death is only 10 to 20 years after symptoms first appear. Tourette's. This is involuntary body movements like motor tics and the use of inappropriate, unnecessary sounds or words. So vocal tics. We don't know what causes it, but it involves a dysfunction somewhere in that neural circuits between the corpus striatum and the prefrontal cortex, just based on the types of movements or vocal that is coming out. You can kind of figure out where connections are a problem somewhere within the brain. Schizophrenia. This is from too much dopamine in the brain that causes delusions, disorientations of reality, paranoia, and hallucinations. Obsessive compulsive disorder. These people have repetitive thoughts, these obsessions, that are causing repetitive behaviors that they feel obligated to perform. So, the example from your book is that thought of somebody breaking into their house, so they have to lock and unlock that door 4 or 5 times before they can leave. Anytime they touch something, they have to wash their hands. That's another obsessive compulsive type of behavior. The cerebellum. That cerebellum helps you learn and it helps you to perform rapid, coordinated, highly skilled movements. So, your cerebellum is going to monitor intentions for movement. So, anytime any kind of plan for a movement is traveling down from that motor cortex, it is going to go into the cerebellum so it can monitor what is happening. It will also monitor actual movement. So as you are moving, this information is coming back up into that cerebellum. So those proprioceptors from your joints and your muscles are sending this information into your cerebellum so it knows how your movements are going. It's going to compare what was sent out with that sensory information that came back up from that actual movement. So it's making sure what was sent down is actually happening. But it also sends out corrective feedback. So if there's any kind of discrepancy with what was intended to happen and what actually happened, that cerebellum is going to correct this information. It's going to help decrease errors and it's going to help smooth out motion. So this information is always going into that cerebellum. It can go back up to that motor cortex. So this information that's going out is correct. And then that sensory information is always going to go back into that cerebellum so it can monitor the motion in as it happens. Circadian rhythm. This is a 24-hour cycle that is your body follows that is set up through the suprachiasmatic nucleus of the hypothalamus. This works with the reticular activating system that's going to help send out all of these different impulses throughout the cerebral cortex. So, increased reticular activating system means we're awake. Decreased in that reticular activating system is what allows us to sleep. Arousal is that awakening from sleep. By having increasing activity out of that reticular activating system, that's what helps to wake us up. There is different sensory stimulus that can activate that reticular activating system, like pain. You ever slept and felt something painful somewhere when you moved in your sleep? That can wake you up. Touch, pressure on the skin, that's why somebody touches you, it can wake you up. Movement of your limbs can wake you up. Bright light, buzzing of an alarm can wake you up. The interesting part is there is no input from the olfactory nerve. So cranial nerve 1, it does not go through the reticular activating system. So the sense of smell does not go through that reticular activating system. This is one reason why we have smoke alarms in our homes. We cannot or detect any smells while we are asleep. Sounds can wake us up, but smell does not. So a smoke detector can take that information and turn it into a sound that can wake us up. So once that reticular activating system is activated, that cerebral cortex is activated, and that's what causes us to wake up, which is what causes that consciousness. Sleep. Sleep is that state of altered consciousness or partial unconsciousness where an individual can be aroused. Why it's important to sleep even during finals week. Sleep deprivation impairs attention, learning, and performance. Not getting enough sleep is going to affect how you do in classes, on exams. So, normal sleep, we have this non-rapid eye movement and this rapid eye movement. We have different stages to our sleep. Stage 1 is that transition from being awake to being asleep. It can last anywhere from 1 to 7 minutes. I would love to find someone who can go through stage 1 of their sleep. It takes them less than a minute or like a minute to fall asleep. It's kind of rare. Stage 2 or light sleep. People can be awoken pretty easily. You may get like little bits of dreams that happen here. Eyes may roll from side to side during this light sleep. Stage 3, moderately deep sleep. Your body temperature and blood pressure decrease at this point. It becomes more difficult to wake somebody, and it actually only occurs about 20 minutes after you fall asleep. And then we have stage 4. This is technically the deepest level of sleep. Your brain metabolism decreases and body temperature drops. It's very difficult to wake somebody. Stage 4 of sleep is when sleepwalking occurs. I used to sleepwalk when I was a child. Some people who do sleepwalk, it's happening during stage 4 of sleep. Test question. So during this non-REM sleep, your blood pressure, your heart rate, and your respiratory rate are all going to drop. Your muscle tone decreases. We have some muscle tone, so we can change positions, but dreams sometimes happen here. They're less vivid though, less emotional, and they're more logical. They make sense. REM sleep, on the other hand, REM, rapid eye movement, eyes are moving back and forth, also known as paradoxical sleep. They can take EEG readings, and it shows very high frequency, small amplitude waves. So, brain blood flow and oxygen use are higher during REM sleep. It is very difficult to wake somebody up when they're in that REM sleep. So here's interesting, their heart rate, respiratory rate, and blood pressure go up. Somatic motor neurons are inhibited. Means we have a significant decrease in muscle tone and paralysis of skeletal muscles, except for the eye, which means during REM sleep, your body can't move. Most of your dreaming is going to occur during REM sleep. These are where there's very vivid dreams. So the visual association in the limbic center. We have decreased activity from the prefrontal cortex at this time. So we go through intervals of REM sleep and non-REM sleep. We alternate between them. When a person first falls asleep, they're going to go through all the stages. It takes about 45 minutes into going through sleep. They may go through that non-REM sleep in reverse order in about that same amount of time before entering that REM sleep. And then that cycle is going to repeat. So during 8 hours, a typical 8 hours period, there's 4 to 5 REM to non-REM cycles. Non-REM, that hypothalamus and that basal forebrain is going to be working. That REM sleep is that pons and that midbrain. Sleep is thought to be brought about by sleep-inducing chemicals within the brain. Adenosine will start to accumulate during high ATP use. Adenosine, that adenosine triphosphate from ATP, we're breaking down ATP, we're left with that adenosine molecule. We get a bunch of it that accumulates. It can start inhibiting the neurons of the reticular activating system that can cause you to feel sleepy and fall asleep. On the other hand, caffeine and theophylline, which is a chemical, caffeine, and then theophylline is a chemical in tea. These block those A1 receptors that prevents that adenosine from binding. And this is what makes caffeine keep you awake at night, or keep you awake, not at night, during the day. So just some interesting facts there on how caffeine actually works. Sleep is essential for your body to function normally. Sleep deprivation. Not getting enough sleep, you can't pay attention in class. Your memory. You actually convert your short-term memory to long-term memory when you sleep. When you're sleep deprived, it's harder to recall information. Another reason why sleep is so important. Your performance declines. Not just grades, physical performance. That's going to decline. You're not going to be able to do certain activities as well when you're more tired. And the more tired you are, the less sleep you get. Your immune system isn't as active, which is what can cause you to get sick easier when you are sleep deprived. If you're not getting enough sleep for long enough, mood swings, hallucination, and even death can occur. Your body needs that sleep to recover and be prepared for the next day. A coma. It's a state of unconsciousness. An individual has little or no response to stimulus. So there can be head injuries, damage to that reticular activating system, brain infection, alcohol intoxication, and drug overdose can be just some of the causes of a coma. If that brain damage is minor or reversible, that person will come out of the coma and recover. But if that brain damage is severe, they may never recover. And that's when these people can go into this persistent vegetative state. So a few weeks of being in a coma, some patients will enter this persistent vegetative state. They have normal sleep-wake cycles, but they do not have awareness of what is happening around them. They cannot speak. They cannot respond to commands. They may smile, laugh, or cry, but they don't understand what these actions actually mean. They're not considered brain dead. To be considered brain dead, there's several different criteria, but one of the criteria is an EEG. If their brain still has waveform, they are not considered brain dead. Brain dead means they have no waveform. Learning and memory. Learning is that ability to acquire new information. We have associative learning, where we make a connection between two stimuli, Pavlov's dog ringing a bell and providing food. That dog soon learns that anytime it hears a bell, it's going to get food. We have non-associative learning. So we need repeated exposure to cause a change in behavior. So habituation, repeated exposure to an irrelevant stimulus causes a decreased behavioral response. So your book's example, a loud sound the first time made me make you jump, but after hearing that sound several times, you no longer respond to that loud sound. Sensitization, there we go. Repeated exposure to this noxious stimulus can increase response. You hurt your arm doing something, that withdrawal reflex can become more exaggerated because you're trying to prevent damage from happening again. So there's different types of memory. So that memory is where that information is acquired through learning, stored, keyword, and retrieved. Just because we store it doesn't mean we can retrieve it. So declarative memory. These are memories of experiences such as facts, events, objects, names, and places. We need conscious recall and is stored in the association areas of the cerebral cortex. A lot of this class has been declarative memory, and it's a lot of facts. Facts require lots of repetition to remember. We have procedural memory, motor skills, procedures and rules. So those motor skills, learning how to walk, run, skip, jump, those are all different procedural memories. Short-term memory. This is a temporary to remember a few pieces of information. You can remember it for seconds to minutes, but it's very limited capacity. And long-term memory. It lasts days to years, but we are only able to recall about 1% of what is stored in your long-term memory. Your memory gets consolidated. So, short-term memory can get transformed into long-term memory when you're sleeping. Your hippocampus is going to play a role. Your hippocampus can be a temporary storage site for this information, but then it can transfer these memories to the appropriate areas in the brain for permanent storage as we need it. So that plasticity for an experience, something to be put into that long-term memory, it must produce persistent structural and functional changes that represent the experience in the brain. Changes in individual neurons, as well as changes in the strengths of synapses or synaptic connections among neurons. Amnesia. It is a lack or a loss of memory. It can be partial, it can be a total loss. We have anterograde is memory loss for events that occur after the trauma or disease that caused the condition. So they cannot form new memories. If you've ever watched the movie 50 First Dates, that movie was based off of a main character with anterograde amnesia. She could not form new memories. And then we have retrograde. Retro always means past. So they cannot recall past events that happened before that trauma. We mentioned language areas in chapter 14, but that language, vocal sounds and symbols that convey information. It can be spoken, it can be written. We mentioned Wernicke's and Broca's area are the two language areas in the brain. They are usually in that left hemisphere only. So Wernicke's, it's that association area that interprets the meaning of spoken words. It can take words and translate it into those different thoughts. So it's going to take in that information and process it. Broca's area. This is your motor speech. It's in that frontal lobe, so it's in that motor area. So it takes your thoughts and allows it to become words. It activates the muscles needed to form the words you want to say. That comes out of Broca's area. So if that word is written or you hear it, Wernicke's area is going to take in this information, process it, and let you know what it means. To respond to somebody's question, it goes into that Wernicke's area. Wernicke's is going to send information to that Broca's area to allow you to form words to respond, to give an answer to whatever question was being asked. Aphasia. is the inability to use or comprehend words. So we have expressive aphasia, damage to Broca's. So they're not able to form words. They know what they want to say, they just cannot get it out. Receptive aphasia, Wernicke's area is damaged. They're not able to understand what they're reading or what they are hearing. These people can speak, no problem, but they can't form a sentence because this information that should be coming in from Wernicke's area to Broca's area does not work. So their sentences sound like a word salad. It's just a bunch of random words because they cannot string comprehensible sentences together. This is where we get word deafness, where they cannot understand spoken words. And then we also have word blindness, where they're unable to understand written words. Most of the time, most aphasias are usually due to a stroke in the brain. So blood vessel blocks, or a blood clot blocks a blood vessel to that specific area in the brain, and that area kind of dies out. So these people are unable to understand what they are hearing, seeing, or both, or they're unable to form words. Parkinson's disease. This is a progressive loss disorder in that central nervous system, usually in that brain. Usually appears around 60 years old, and it involves the substantia nigra in that putnam and caudate nucleus. And it has to do with the neurons that release that dopamine degenerate. They break down. So we're still, those people, they still receive acetylcholine, but there's not enough dopamine in the brain. And this is what causes most of the symptoms. We don't know for sure what causes Parkinson's, but toxic chemicals, pesticides, herbicides, carbon monoxide can contribute. Only 5% of Parkinson's cases have shown to be hereditary. So these people, you see a lot of involuntary skeletal contractions that interfere with their movement. They have tremors. This is what most people see when they think of Parkinson's. They have this tremor. So their hands, when they're trying to do something, shake, which makes it hard for them to be able to actually perform certain movements. They also have rigidity of their facial muscles. They're contracted, they're stiff, they're kind of holding these contractions. They have bradykinesia, slower movements, and they have hypokinesia, a decreased range of motion. Since they have this very low levels of dopamine, treatment includes increasing their dopamine levels and decreasing those acetylcholine levels, because that large amounts of acetylcholine actually causes toxicity within the brain. Big problem is because since this is within the brain, is crossing that blood-brain barrier to get medications into the brain is very difficult. There is a drug that is called levodopa or L-dopa, and it works to relieve some of the symptoms, but it doesn't slow the disease process. They're still working on trying to find treatment to help actually slow the progression of Parkinson's. But as of at least when your textbook was written, nothing has come out specifically for like actual treatment that helps to slow the progression of Parkinson's. And that is it for chapter 16. Thank you very much for listening.